Hello, everyone. Welcome to Scholar Labs, a one of a kind platform that digitizes science experiments with immersive 3D augmented reality. Our engaging simulations, developed by experienced teachers, enhance learning and are accessible on smartphones, tablets, laptops, and desktops. Today, I will be taking you through the simulation life processes. This simulation encompasses various key biological processes taking place in the human body. Before starting the simulation, let's get familiar with some important control buttons. This I button on the top right corner of the screen will take you through the objectives of the simulation and also guide you through it by giving step-by-step instructions on how to run the simulation smoothly. These buttons on the right side of the screen will allow you to view the simulation from different angles. Use this mouse scroll button on the bottom left corner of the screen to zoom in or zoom out. Tap on this bulb icon to switch on or switch off the contextual help. So let's begin. There are certain vital processes which work together for proper functioning of the body. They are called life processes. These processes are essential for all living organisms, including plants and animals. These life processes are nutrition, respiration, transportation, and excretion. Click on nutrition to know more about it. The process of acquiring food that is needed for nourishment and sustenance of the organisms is called nutrition. You can see two main modes of nutrition on the right side of your screen. Autotrophic nutrition is present in plants which produce their own food using light energy through photosynthesis. Refer to autotrophic section in the simulation Nutrition in Plants for detailed information on this topic. Click on heterotrophic nutrition to view how amoeba derives its nutrition. Click on the dropper to take a culture of amoeba from the jar and drop it on the slide. Click on the slide to cover it with the cover slip and place it under the microscope. Click on this eye button to view the feeding in amoeba under the microscope. Click here to pause the scene. Amoeba engulfs food using temporary finger-like projections of the cell surface which fuse over the food particle, forming a food vacuole. Inside the vacuole, complex substances are broken down into simpler ones, which then diffuse into the cytoplasm. The remaining undigested material is moved to the surface of the cell and thrown out. Now take a quiz to test your understanding of the topic. Click on the right answer and then click on Next. To go to the previous question, click on Previous. Click on the Submit button to check your answers. If you wish to give it another shot, click on retry, otherwise, click on finish. Tap on respiration to understand the respiration process in organisms. Respiration broadly means the exchange of gases. Animals and plants have different means of exchange of gases. To know more about respiratory system, kindly refer to the simulation respiration. Click on the lime water experiment tab to compare the carbon dioxide exhaled by humans and an instrument like syringe through an experiment. Click on the test tube to make the boy blow out air into lime water using a straw. Observe the time taken for lime water to change color and turn milky. Now, click on this straw to connect it to the bulb syringe and place it inside the other test tube. Click on the bulb syringe to pump air out into the lime water. Observe the time taken for lime water to change color. With this experiment, we can conclude that humans exhale more carbon dioxide as lime water turns milky much faster, that is in 7 minutes 30 seconds, than passing air through the bulb syringe, which took around 13 minutes and 20 seconds to do so. Now, click here to see how lung capacity varies in different individuals. Click here to blow air into the balloon multiple times and observe the lung capacity in different individuals. From here, you can conclude that tuberculosis patients and asthma patients have low lung capacities. This is because they have restrictive lung conditions and their airways don't expand fully, while a middle-aged person has the highest lung capacity. Now let's understand what is cellular respiration. Cellular respiration is a set of metabolic reactions occurring inside the cells to convert the energy obtained from the food into a chemical compound called ATP, that is adenosine triphosphate. Click here to start the running action. You can also increase the speed of the runner by clicking here. Observe the energy level. Manage the energy level of the runner by providing food at regular interval. Click here to select any of the food items. Click here to closely observe the cell structure. With Scholar Virtual Lab, students can visualize the process of cellular respiration, making it easier for them to grasp the flow of energy. Click on the cell and experience cellular respiration in the virtual world. The first step in cellular respiration is glycolysis, which occurs in the cytoplasm of the cell. In this, glucose undergoes a series of chemical transformations and gets converted into two molecules of pyruvate. In this process, two ATP molecules are produced and NADH is released to be used in later steps of cellular respiration. Stage 2 is the preparatory reaction. During this stage, the pyruvates of glycolysis move into the mitochondria where They are converted into acetyl coenzyme A. During this conversion process, more NADH is produced and carbon dioxide is released. In stage 3, the citric acid cycle, the remaining carbons from the initial glucose are oxidized, releasing carbon dioxide. NADH and FADH2 are also produced in addition to two ATP molecules. Most ATP production occurs in stage 4, the electron transport chain or ETC. In this stage, the NADH and FADH2 from the previous stages give up electrons to the chain. Energy is released and captured as the electrons move from a higher energy to lower energy state using a series of proteins embedded in the membranes of the mitochondria. Later, this energy is used for the production of ATP, typically 32 to 34 per glucose molecule. Oxygen is used by the ETC as a terminal electron acceptor. It then combines with hydrogen ions to produce water. If we add up all the ATP produced from just one glucose molecule, we can see that these reactions produce a total of 36 to 38 ATP. Click on the cross button to go back to the main screen. Now let's observe what happens if we do not replenish his energy. Click on the start button. This time, do not select any of the food items. Keep an eye on the energy meter. You can see that the energy level of the runner is dropping significantly. While performing vigorous physical exercise, there is deficiency of oxygen as it gets used up continuously. Due to low level of oxygen, anaerobic respiration occurs in the muscles. As an end product of cellular respiration, lactic acid is produced which gets deposited in the muscles. Lactic acid deposition leads to muscle fatigue, causing pain, and the muscle fails to contract further properly. The next life process is transportation. All living organisms need a few necessary components like air, water, and food for their survival. These elements are transported to their body cells and tissues by a transportation system. In plants, the vascular tissues are responsible for transporting substances, while in humans it is done by circulatory system that consists of blood, blood vessels, and the heart. For the topics circulatory system, blood constituents, and transportation in plants, refer to the simulation Transportation in Plants and Animals. Click on this tab to learn how to measure blood pressure. Read these guidelines very carefully and follow these steps. Let's take the case of a 25-year-old girl. Click on the BP cuff to wrap it around the upper arm of the girl and place two fingers on the inside of her wrist to listen to the pulse. Click on the inflation bulb of sphygmomanometer to pump up the cuff slowly and then listen to the pulse. Stop inflating the cuff when the pulse disappears. Start to deflate the cuff slowly whilst watching the mercury level in the sphygmomanometer. Note the reading when the pulse reappears. This is recorded as the systolic pressure. Deflate the cuff further until the pulse disappears. This is recorded as diastolic pressure. Now, click here to take a quiz. Choose the correct option and then click on Next. To go to the previous question, click on Previous. Click on Submit to see your answers. Scholar Virtual Lab offers immediate feedback on students' actions helping them understand their mistakes and learn more effectively. So, do not shy away from taking the quiz one more time by clicking on retry button. Otherwise, click on finish. Another important life process is excretion. It involves removal of metabolic waste materials and other non-useful substances out of the body. To understand the excretory system in humans, refer to the section Excretory System under the simulation Transportation in Plants and Animals. Click on this tab to explore the 3D model of kidney and the working of nephron in human excretory system. The excretory system in humans includes a pair of kidneys, a pair of ureters, urinary bladder, and urethra. Press down the left mouse button and drag it to have a 360-degree view. Each kidney has millions of nephrons. Click on the left kidney to expose its internal structure. Again click on it to explore the structure and functions of nephron. Each nephron is made up of two parts, a cup-like structure known as Bowman's capsule and renal tubules. The Bowman's capsule encloses a bunch of capillaries called glomerulus that filter waste materials from blood. The renal tubule has regions called proximal convoluted tubule, loop of Henle, and distal convoluted tubule. These regions absorb useful substances back into the blood and filter the remaining waste substances. The output from nephron is called urine. Thanks for watching.